Hemoglobin and myoglobin. Molecular oxygen plays an important role in biological system because of its importance in respiration. The formation of oxygen we have already learned in nature involves a number of reactions such as redox reactions, electron transfer, atom transfer, and photochemical reactions. A number of proteins are known that bind and transfer oxygen to different extent. Such proteins that bind oxygen are called oxygen carriers. The most important and well documented proteins that bind and transfer oxygen are hemoglobin and myoglobin. Hemoglobin and myoglobin are very closely related to each other. Hemoglobin is a larger protein with a molecular mass of 64,500 and consists of 4 subunits. Each subunit is made up of a heme group associated with a protein called the globin. It is important to recall that each heme group is composed of an Fe2 positive ion surrounded by 4 nitrogen of a porphyrin ligand. Hemoglobin is a large intercellular protein that is responsible for the red color of the blood cell. On the other hand, myoglobin is relatively small protein with a molecular mass of approx 17,000 and consists of one heme group and a polypeptide chain. It is important to recall that the peptide chain consists of 150 to 160 amino acids residues folded about the single heme group. Myoglobin is a small intercellular protein in vertebrate muscles. Myoglobin was the first protein whose X-ray structure was established in 1959. In 1962, the Nobel Prize in Chemistry was awarded to Kendrew for their work on the structure of globular proteins, including myoglobin. The basic functions of hemoglobin and myoglobin are the following. Hemoglobin picks up the oxygen in the lungs and transports it to the rest of the body. Myoglobin accepts oxygen from the hemoglobin in the muscle and stores it until needed for energetic processes. Deoxygenated hemoglobin uses some of its amino acids to fix up CO2 and then transport CO2 back to the lungs. Oxygen carriers perform the function of acquiring O2 from the atmosphere, transporting it through the circulatory system of the organism, and delivering it to the location where it is used. They all bind O2 rapidly and reversibly according to the equation protein plus O2 gives you protein O2. The equilibrium must be largely to the right at the oxygen concentration available at the pickup location. For example, lung air interface, muscle cell blood interface, mother fetus interface at the placenta. It is important to note that the oxygen carriers interact strongly with oxygen to bind it. However, the carriers must not undergo irreversible oxidation in the process. In brief, hemoglobin picks up oxygen in the lungs and transports it to the site of use inside the muscle cells. This oxygen is transferred to the myoglobin, which stores it until required for metabolic action, in which O2 is reduced to CO2. Deoxygenated hemoglobin then transports CO2 back to the lungs. Structure of hemoglobin and myoglobin. We have already mentioned that the structures of hemoglobin and myoglobin are very closely related to each other. Hemoglobin is a large protein and consists of 4 subunits. Each subunit contains one heme group associated with protein globin. The iron is held in position by coordination with the nitrogens of heme and an imidazole of globin complex. This complex reversibly with oxygen. Thus, hemoglobin is the oxygen carrier of blood, and the red color of blood is due to the oxygenated hemoglobin. It is important to note that the four site units are similar but not identical. The two subunits from alpha chain of hemo F141 amino acid, while the other two subunits from beta chain of 146 amino acids. These chains are coiled together to give a three-dimensional network, a systematic structure of hemoglobin showing the relationship among the four subunits of the tetramer is shown in the figure. Structure of hemoglobin, myoglobin. Myoglobin is a small protein compared to hemoglobin. It consists of a single heme group embedded in a polypeptide chain called globin. The peptide chain consists of 150 to 160 amino acids folded about the nit- single heme group. The heterocyclic ring system of heme consists of a porphyrin containing pyrrole groups labeled as A, B, C, D linked by methane bridges. The iron atom in the oxidation state 2 is present at the center of the heme group, is bonded by four nitrogens of the porphyrin system together with one nitrogen atom of the imidazole side chain of the histidine residue, which is part of long protein chain of amino acid residue. It is this polypeptide chain that plays an important role in the biological function fixation of molecular dioxygen. A simple outline of the structure of myoglobin showing heme group embedded in the polypeptide chain. It is important to note that both hemoglobin and myoglobin have five coordinated iron atoms. The oxidation state of iron in Hb and Mb is +2. Iron atom is bonded to four nitrogen atoms from pyrrole ring and the fifth nitrogen from the protein chain. In both the compounds, the sixth position is occupied as weakly bonded to water. Hemoglobin and myoglobin in such molecules are named as deoxyhemoglobin and deoxymyoglobin, respectively. However, when molecular oxygen occupies the sixth position, which is trans to histidine chain, then the molecules are named oxyhemoglobin and oxymyoglobin, respectively. Myoglobin polypeptide is comprised of 8 separate six-handed alpha helices designated from A to H. They are connected by short non-helical regions. Role of hemoglobin and myoglobin. Hemoglobin and myoglobin plays crucial role in transporting dioxygen O2 from lungs to tissues and CO2 from tissues to the lung. The function of hemoglobin is to bind O2 at the high oxygen partial pressures found in the lung tissues to form HbO2. Hemoglobin carries this oxygen without loss through the blood and then releases it to the myoglobin. In other words, the dissociated oxygen from hemoglobin diffuses to the tissue where myoglobin picks it up and stores it until it is needed. It means that myoglobin has greater oxygen affinity than hemoglobin at low partial pressures. The hemoglobin and carbon dioxide are then returned to the lungs where CO2 is exhaled. It is clear from the scheme too that both hemoglobin and myoglobin play cyclic role in transporting O2 from lungs to the tissues and CO2 from tissues to the lungs. The CO2 is carried out in the form of HCO3 negative ion. Careful analysis of the scheme suggests that the partial pressure of O2 is high in the lungs. This is responsible for the formation of HbO2. However, the low partial pressure of O2 in the tissue is responsible for the dissociation of HbO2. The dissociated oxygen O2 diffuses into the tissues and oxidizes the metabolic fuels to CO2 and H2O. The carbon dioxide evolved is exhaled in lungs. Oxidation and oxygenation of Hb and Mb. The heme group consists of Fe2 positive ion embedded in a porphyrin ligand. The Fe2 positive ion has 6 coordination sites. The porphyrin ligand takes up only 4 of the 6 sites, leaving 2 free binding sites on opposite sides of the metal ion. If a free heme is present in aqueous solution, the 2 vacant sites may be occupied by water molecules. In an aqueous solution containing oxygen, a free heme group binds to oxygen molecule in an irreversible manner with the oxidation of Fe2 positive to Fe3 positive ion. The oxidation of iron from Fe2 positive to Fe3 positive destroys the oxygen binding activity of iron. This unproductive oxidation in a biological system is potentially fatal. The irreversible oxidation of heme in the presence of oxygen involves a series of dimerizations containing two Fe ions, FeO, Fe. The dimer is known as hemetin. This dimer can no longer act as oxygen carrier. Deoxyhemoglobin is a 5 coordinated high spin complex T2G 4Eg2 with 4 of the coordination sites of iron being occupied by 4 nitrogen of the porphyrin link, while the fifth coordination site is occupied by the nitrogen atom of the imidazole ligand of histidine residue which joins the heme with the protein. In the absence of O2, the ligand is weak and have 5 coordinated heme complex of Fe2 are always high spin. The diameter of high spin T2G4 Eg2 iron ion is larger than that of the central hole of the porphyrin ring. The estimated Fe-N bond distance is about 218 pm in high spin Fe complexes, while the size of the hole of the porphyrin ring is about 200 pm. As a result, the iron ion is compelled to 0.40 Armstrong above the plane of the ring formed by the 4 nitrogen atoms to give a square pyramidal arrangement, thus distorting the bonds around the iron. When the oxygen bond at the sixth position of iron opposite to the histidine, the iron ion contracts and move into the plane of the ring. In other words, Fe2 positive ion becomes coplanar. This happens because the moment oxygen gets coordinated to iron, the ligand field becomes strong, which results in spin pairing. You recall the spin pairing always lead to low spin complexes. In this case, the electronic configuration of iron changes to T2G6 and the resulting complex is diamagnetic. The Fe-N bond distance is approximately 200 picometer, which matches with the size of the whole of the porphyrin system as shown. It is important to note that the binding of molecular dioxygen to the sixth position of iron atom in the deoxyhemoglobin and deoxymyoglobin releases the strain in the 5 coordinated system. This release in the strain is compensated by the loss of spin pairing energy is going from 5 coordinated deoxyhemoglobin or deoxymyoglobin to 6 coordinated oxyhemoglobin or myoglobin respectively. It means that the coordination of O2 will cause Fe atom to drop into the plane of the heme group. The shrinkage in the size of Fe2 positive ion and dropping into the plane of the porphyrin ring is shown. The dropping of iron ion into the plane of the porphyrin results in the perturbation in the imidazole group of the histidine residue present at the trans position. In other words, the decreased size of the iron ion is sensed by the imidazole group of the histidine residue, which behaves like a spring. In doing so, Fe2 moves the histidine molecule and causes conformational changes in the globin chain. Since the chain is hydrogen bonded to the other three units, it changes their conformations as well, and the net effect of this interaction and in hemoglobin is the increase in the ability to attract O2 of the second heme site and so on. It means that the one iron binds an oxygen molecule, the molecular shape changes to make the binding of additional oxygen molecules easier. Since each heme group is in hemoglobin can bind one oxygen molecule additional, the process continues till it carries a maximum of 4 molecules of oxygen. This phenomena is called cooperative effect or autocatalytic nature of O2 binding. Myoglobin and hemoglobin functions and cooperativity. The function of hemoglobin is to bind molecular oxygen O2 and the high oxygen partial pressures found in the lung tissues and then carry it with loss through the blood to the tissues where myoglobin picks up O2 from hemoglobin. The sequence indicates that myoglobin has greater O2 affinity than hemoglobin at the low partial pressures of oxygen.